Now we know it's happening. All right. Hello, everyone. Welcome to our community research update call. I'm Lauren Beauregard. I'm the incoming executive director. Let's see. Thank you all so much for tuning in, whatever time it is, where you are. Wanted to let everybody know really quick, we are recording this session for posterity. And so just, I want everybody to be aware of that. The only other thing we want everybody to do is please leave yourself muted while our presenters are speaking. We're going to have time for questions at the end. If you think of a question, you want to make sure that you don't forget it or that it gets answered, you can drop them in chat while people are speaking. But otherwise, hold your questions and we'll kind of use the raise hand feature and call on people that way, or we'll read your questions from chat. You're welcome to enter them during the questions section as well. All right, so with that, we're gonna go around, it sounds like, and introduce ourselves. So I just did. So I'm gonna hand it over to Noreen. We'll popcorn style it.
Hello, thank you. While I'm doing my introduction, Hannah, do you want to bring up your slides? I'm the collaborative research coordinator for CLOVES. Very excited for this call and for to hear a little bit more about the work that these wonderful researchers are doing. And once Hannah, I will have her kick us off for our introductions.
Hannah, you're muted. I've just pressed.
Thank you. Sorry about that. It's been a long day. Thank you so much for inviting me here today. I'm Hannah and I'm working for Liz Patton at the University of Edinburgh, and I'll be talking about zebrafish later on. Who wants to go next?
Ralitsa, you're unmuted. You go.
Oh, hi everyone, I'm Ralitsa. I am a PI or principal investigator, lab leader at the University of Dundee. I used to work with Rob Semple as a PhD student, and that's how I got into PIK3CA related overgrowth research. And I think now it's the time to hand over to Rob on that note.
So hi, yeah, I'm Robert Semple. I'm a doctor and a scientist in the UK, and my group was one of the first groups 10, 12 years ago to find PIK3CA mutations, and I've met lots of fantastic people since then with CLOVES and similar syndromes. But my major contribution has probably been to try to entice talented younger scientists into the field to move things on, and Ralitsa is one of those. Liz, would you like to go next?
Sure. My name's Liz Patton, and I'm a professor at the University of Edinburgh. And I work with Hannah and also with Rob and Ralitsa as collaborators. And I'm quite new to the CLOVES community, funded through CLOVES. We started a project in our lab last year, and that's what Hannah will present today. And it's been a great pleasure and privilege to be here today.
I think that's everyone. Ralitsa, go ahead, kick it off.
So if that's all the introductions, could you go into presenter mode, please, Hannah, and move on to the next slide? So there you go. There we all are. You've heard who we are. For those who are across the ponds, just to explain where exactly we are, we're in the UK, and we're in Scotland, which is the top bit of the UK. And we're in these two cities here, Edinburgh and Dundee, which I can tell you are very cold and chilly today. So it's lovely to be here, and what we want to do is just give you a little bit of an update on some of the research that we're doing, and that's research which has only been possible because of the support that we've had from the CLOVES syndrome community. And we want to explain what we're doing, and we also want as many questions as possible, please. And there really is no such thing as a question that's too simple. And we do our best to make our very technical language easier to understand, but sometimes we don't get that right. So please don't be put off if you see any very technical words. Please ask any questions that you want at the end. So it's my job really just to start off and introduce some of the fascinating work which Hannah and Ralitsa have mostly been doing. And so I really don't need to tell you what Clove syndrome is, and I'm only using this slide to remind everybody that it's really useful, the name Cloves, but of course there are lots of other conditions which look quite like Cloves, but might not exactly meet the criteria for Cloves. And they could have their own name. They could be KTS, Klippel-Trenaunay syndrome, or they might be a range of conditions which have some of these features that you see here today. And I think it's very important that we remember all of those and we're not exclusive. And so that's why we use the language of Cloves or prose to cast the net as wide as possible, because I think we can learn a lot from the full range of the conditions. So if you go to the next slide, please. Hannah. And there's an awful lot of elements to making sure that everybody with prose and Cloves is well looked after, and medicines are only some of those. But what we're talking about in particular today are how we might learn more about the condition so that we can help predict the future, so we can help advise people as best as we can, and also, of course, to try to find new and better medicines. And when we think about better medicines, they come in sort of different shapes and sizes. The best hope is that through research, we find out that a medicine that is already around, that is already available in hospitals or clinics, works in Cloves. That's called repurposing. Or sometimes we find that there's a medicine that is nearly available in hospitals and is still being researched by companies that make them. And then we try to persuade those companies to use their new medicines for Cloves and prose. But also, of course, we spend a long time looking for completely new ways to treat things. That is, find entirely new things about the way Cloves develops so that we can interfere it better than we can at the moment. And that, of course, takes a long time. It might take 10 or 15 years for that to happen. Whereas if we find a medicine that's already available, that might happen in months or years. And we want to give the benefit of all these possibilities to everybody. So that's really what we're talking about. So next slide, please. And the picture on the left is just simply to explain why it is so complicated to study and understand Cloves. And that's because even though the very same gene changes cause the condition in more or less everybody, every single person is very different. And the exact pattern of problems which every person has is distinct, because it depends when the change first happens and when it happens in the womb. And that's just a sketch of all the different things that happen in the first 7 or 8 weeks of life. And although we can look at those very early stages of life with some scans, we really aren't able to look at that very well. So by the time children are born with Cloves, who go on to develop more features of Cloves, we already haven't been able to see a lot of the really important events which determines what happens later on. So we have to deal with the challenge that it can look very, very different in many, many people. And it takes time to grow as well. So we need to be able to follow things over the long period of time. So if we go on to the next slide, this explains why we talk a lot about models. So when we mean a disease model, we mean instead of studying people, who in the end are by far the most important thing, but who take a long, long time to study, we need to find smart ways to speed things up, because we're impatient, like you're impatient, to learn as much as we can as quickly as possible. So when we say models, we mean cells or animals or something else where we can produce some of the important processes in Cloves so that we can study them quickly, and we can study all the variation as quickly as possible. And so rather than having to wait years and years and years every time we want to see if a treatment works, we can do it over the course of days or weeks. And you'll see 2 examples of ways in which we are trying to do that at the moment. And this is where I'm going to hand over to Ralitsa, who is going to tell you about some of the work which has been funded by the Clove syndrome community starting off with some cells, but I'll hand you over to Ralitsa at this stage.
Thanks, Rob. So before I actually go into it, I just want to say that this should be fairly informal. So I hope everybody's relaxed and not worried about asking questions, presenting, because I can sense that people are a little bit tense. And so just for the record, I've practiced absolutely nothing because today has been a really exciting day in the lab, partly because of the money we've been given by CLOVES syndrome community. We've been able to make some really exciting progress, which means that we've been super busy in the lab and not had enough time to prepare for the presentation. But hopefully you'll be able to follow along. And if there are any questions, please do ask them at any point, because this is really for everybody, for you guys, not really for us. So hopefully I won't be mentioning too much jargon, but there is a high risk that may creep in. So again, just ask for clarification if anything is unclear. Rob already alluded to this, but I think it's really important to emphasize that all models are wrong, but some are useful. And so that is the situation that we're in when we're trying to study PROS or CLOVES. I am on the low complexity, well, I should say not low cost end of the spectrum that you're seeing in front of you. I'm sort of in the middle in terms of the cost. So you can start off with very simple models, cells in a plastic dish, and then you can move towards more complex models. Zebrafish, you'll hear about that from Hannah. You can also have mouse models. I'm sure many of you are following the literature and have come across studies that have been published that, you know, present various bits of evidence that certain treatments are working and others are not. But again, whenever we read these types of papers, it's really important to remember the limitations of all models. And so the model that I am working with is a cell line. So it's a so-called human-induced pluripotent stem cell line. I'll show you in a second why that's useful. And it's somewhere here in the middle. It's fairly expensive to work with, but it has certain advantages that some of the more simple cell models do not have. And certainly it also has limitations which we can address by going to more complex model systems like the ones that Hannah will be talking about. Next slide, please. Now, a lot of what we know about PIK3CA has historically come from cancer, but of course, CLOVES or PROS, they're not cancer. And so we need a better model than cancer cell lines. And when I started my PhD in Rob's lab back in 2014, 15, I was tasked with making the first developmental model system with a PIK3CA defect, that the one that we see in PROS and CLOVES. And so the reason for that is because the PIK3CA defect that is causing CLOVES is acquired developmentally. And so we can't just take a cancer cell line and expect to find something that is of relevance to CLOVES necessarily. So we started off with these pluripotent stem cells. So these are multifunctional cells which can give rise to any other cell type in the body. And we wanted to work with these because in PROS and CLOVES, the tissues or cell types that are affected are very different from the ones that are affected in cancer. So we want to be able to study fat cells or adipocytes, neural cells, muscle cells, liver cells. And the cells that I haven't listed here, but are the ones that I've been working with all day today, are actually endothelial cells, so the cells that are lining the blood vessels, which are often affected in patients with CLOVES and PROS more generally. So we've used a fancy tool, a genetic scissors called CRISPR, which many have heard of nowadays, I think, to introduce the PIK3CA H1047R mutation that some of you will be familiar with into these cells. And now we have access to a rich resource from which we can generate all these other different cell types. Some of the progress that we've made in the last year, thanks to funding from you guys, is with respect to making endothelial cells. So Hannah, next slide, please. Yeah, sorry. With respect to making endothelial cells, so the cells that line the blood vessels. And what you can see here, I don't want you to worry about the labels that are on the top. These are different colors. The different colors are markers for certain, they're so-called molecular markers. So for example, if we're making endothelial cells, we know that they're supposed to be having this purple marker and the yellow marker. And on the left-hand side here, we have cells that are normal. They don't have the PIK3CA defect. So that's this row that you see here. And then we've got cells that have the PIK3CA defect either in 1 copy or 2 copies. So this is a little bit of technicalities, but essentially in pros and cloves, cells that are affected have 1 copy of this PIK3CA defect. And hopefully what you can see here, without, again, paying attention to the labels, is that the pictures that you see in this second row look different to the ones that you see in the top row. And for us, this is really exciting, because this is the first time that we're able to make these cell types with the PIK3CA mutation or defect that is of relevant to your disease, and B, because we can actually see aspects of behavior of the cells that we would have expected based on what we know about the human disease. And so what we can now do is use this rich resource to address so-called mechanistic questions. So that's what my lab is doing. We're really asking nitty-gritty questions about how this whole signaling that PIK3CA is triggering is wired in the cells. And that's really important because that tells us more about the complexity of PIK3CA in the context of the different cell types, and also allows us to look at more sophisticated ways of targeting or treating cells with a PIK3CA defect. Next slide, Hannah. Thank you. And this is really important because for the last 30 years, so PIK3CA, this gene in which there is a defect in cloves and pros, PIK3CA has been known for 30 years. But for 30 years, the understanding that we've had of PIK3CA and what it's causing in cells has very much been one that I call the switch understanding. So people tend to think about PIK3CA defect switching this on. So the switch is on when you have the mutation. And so if you want to treat the disease, you need to switch this off. The problem here is that PIK3CA is important for all cells, not just the ones that have the mutation or the defect, but also all the other cell types that we have in our bodies. And so if you just switch this off, you're going to cause havoc in a lot of cells that actually require PIK3CA to be there. And so if you think of something like alpelisib, although it is helpful, or Piqray, some of you will know it as Piqray or VJoyce, it is helpful for a lot of patients, but at the same time, many of you will know that it can also cause toxicity, and that toxicity is because you switch this off. Now, what we've learned in the last 4 to 5 years is that things are much more complex. Actually, we've learned this already since PROS was discovered. When you look at all the different ways in which cloves presents itself in different people, you know that just because you have the same PIK3CA defect, you're not identical. And so we now know that PIK3CA can present itself in many complex ways. And the way I tend to represent this is with an accordion. So just like the same piece of music can be interpreted and played in different ways, so the outputs of a PIK3CA mutation can be very different depending on the context that we're looking at. So for example, which cell type is it? At what developmental stage was the defect acquired? What's the environment of the cells? And also what is the mutation? And last but not least, we now also think that there are important aspects of what we call intensity, signal intensity and dynamics that are also really important. And this is nitty-gritty details about biochemistry that I'm not going to go into, but essentially something that we're now addressing in the lab because we want to be able to exploit this complexity for therapeutic advantages beyond what is possible simply with alpelisib or VJoyce. Now, I can't deliver on that on my own. We need a collaborative effort. That's why we're here as a team. And ultimately, some of the findings that we observe in my lab will have to be tested in more complex model systems where this is possible. And that's where Hannah and the fantastic zebrafish model that she has made comes in. And so I'll hand over to Hannah now to present some really great progress on that front.
So hello, hopefully this works. So you might be wondering why we were using zebrafish of all things. So despite them looking very different from us, they actually have very similar genes and cells to people. So actually 70% of the genes that we know cause disease in people have a counterpart in zebrafish. And as you can see from this little movie on the left, they develop really rapidly. I mean, obviously this is sped up, but they go from having just 2 cells to an embryo with recognizable eye, tail, and they can move in just 24 hours. So that's great for when we're doing lots of experiments at once and testing lots of drugs, for example. Also good for us is that their genetics, their DNA, is quite easy to modify. So we can do things like introduce the cloves gene to try and mimic in a whole organism like a complex disease. And unlike mammals, they develop externally, so they lay like 300 eggs at a time. And because they're see-through, we can look at them under a microscope and we can watch in real time the very first things that happen when a mutation happens, which we thought made it ideal for looking at Clove syndrome. So we started a collaboration with the Clove syndrome community with Rob, and obviously I work with Liz. And we wanted to help make the first zebrafish model of this to answer some of the questions and to add to what Ralitsa is doing. And you might have seen on the website that we had this this collaboration with the Clove syndrome community, and that really helped us move from just the very, very basic questions. I mean, I came from a slightly different field. I didn't really know what PIK3CA was, but to sort of answer the very basic questions, like, is zebrafish and human PIK3CA similar enough? Is it active in early fish development like it is in people? Is it going to be a good model? And that has enabled me to, answer those today, and today I can show you how we can model aspects of cloves and vascular anomalies in zebrafish. So this is one of the very first little fish embryos that I saw that had a little vascular lesion here, and it was so exciting when I saw it under the microscope. It's like, finally, because there's a lot of blood, sweat and tears that goes on in the middle here where I wasn't sure it would work. So how did I go about this? So to develop a zebrafish model, as I said, we can change the genetics of zebrafish quite easily. So what I did was I injected DNA that coded for the cloves mutation into zebrafish embryos. You can see my needle going in. I took this under the microscope. And what will happen is that as the embryos divide and grow, they will activate the PIK3CA mutant gene in patches. So like in people, it's kind of random where this mutation will end up. So it mimics the random nature of cloves in that sense. And because what I've done is attached to the DNA with the PIK3CA mutant, there's a pink fluorescent protein that is coupled to it. So we can see, as we watch the zebrafish grow, we can see these pink cells appear, and this means that we can actually see the PIK3CA mutant cells and watch what they'll end up doing. And some of the other cool things we can do with zebrafish is that we can look at specific types of cells. So on the left here, this is, I won't say ordinary zebrafish, but actually it has all the blood vessels it develops and grows are glowing green. So when we look under the microscope, we can see all these little blood vessels develop and form. And I noticed that when we inject our PIK3CA mutant gene, this is kind of messed up. So this is one example here. So the fish is on its side is zooming in. So I noticed that there's a big difference between these two vessels here, and we get this kind of too big. And what was interesting is these PIK3CA mutant cells, we can see they're not within the lesion, but they're touching it. And similar to here, this embryo had a bit more of a serious lesion. And here we've got the equivalent vessel here that was quite narrow, but it's really, really big and overgrown in this sample. And what we found really exciting about this is that we've got some blood vessel cells that are green, and they're sort of genetically normal. But these are sometimes separate from the PIK3CA mutant cells. So we think what's happening is maybe these PIK3CA mutant cells are able to communicate and maybe send messages to the healthy cells to cause them to overgrow as well. And that's something really exciting that we're hoping to follow up. And in addition to being able to model the vascular problems, I've also been growing up some fish a little bit older. So here, 2 weeks old, we've got they're free swimming, they're eating. I noticed although this fish was happily swimming around, it had a kind of wobbly appearance on the back. And I think this is because some of the muscle tissue is overgrown. And similarly, I can also stain for the bone and cartilage of the fish. And compared to this nice, like regular structure of the spinal column at 3 weeks old, I notice in some of my cloves fish that I get thickened vertebrae. So I think something else is going wrong, either with the sizing here or with the timing of how the bone is forming. So this shows that we can start to mimic just several different aspects of close and pros, which hopefully will be really useful. So what I'm hoping now is that we've got this zebrafish model and we can use this as a springboard to be able to really get to grips with the imaging, the different stages of pros, like in real time in a whole animal. And we can follow up, how does pros change healthy cells? How does the Clove syndrome, corrupt any healthy cells? And hopefully this means that we can be better informed when we're looking at drug screens, which we can also quite easily do in zebrafish, and to find new and better therapies that will really help individual patients. So I think that's all that I wanted to say. So if Rob, or Ralitsa, do you want to say the last slide?
Yeah, I'll maybe come in. Thanks very much. So I'm really hoping you're going to pick us up with lots of questions about all the sort of jargon you've heard along the way. But I think that's a beautiful demonstration of how we can go back to what we want to do at the start. We want to either find the best way to use the medicines we have or find completely new medicines. And with a medicine we have, like alpelisib, Piqray, Rejoice, we can test that very quickly in a fish at lots of different stages to find out if there's one particular stage when it has the most benefit. Or we can try to hunt down these signals that we think the bad cells use to make even the good cells grow, and maybe find a completely new way of blocking things. So that's why we're excited about these. And what we will try to do is to use the fish studies together with the human cell studies to try to work out as quickly as possible what the best bet is for new treatments, which we can go and try in human beings. And of course, we want to back the right horse, if you like, as early as we possibly can, because I think we all know from the trials which are underway at the moment how long it can be to prove something in people. And we think these model systems are very powerful in combination to do that. So if you can just give the very last slide, I think there's just one more slide, and then go through the animations. And so I can't emphasize enough how important the support of the CSC has been in allowing us to get this far. It's also true that patient groups, patient charities can't afford to fund all the work that's needed, but what they're so fantastic at doing is, first of all, allowing us to do enough work to persuade other funders to put more money into this area. And we're very hopeful that that's going to be successful this year. But it's also very important to have the backing of people who are affected by these conditions and advocacy groups and charities, because all our research funders really expect us to show that we're working together in partnership. So the old days of scientists and doctors doing clever things on their own and telling you the answers are long gone now, and that's all for the best. We need to show that we are listening to what's needed most and using that to work out how to do our research. But it's not always about going for the quick win, the magic drug or the first thing that we can find. We also need to be patient, and we need to bear in mind that it's crucial to look after the people that do the research. And doing science is a tough career because you keep having to raise money to do your next bit of work. And this is very difficult often for young, talented scientists coming through to keep going it. So other great uses of money from organizations such as the Clove syndrome community are to make sure that younger scientists keep going at points where they need a little bit of extra time to get the next grant. And also to make sure that they're using their funding to ensure that everybody is working together for the common good. And doing it in the way that it's intended, so that all scientists around the world and all doctors can see the results, so that we get the best results as quickly as possible. And that's just what this slide captures. So I think we should come out of presentation now and hopefully be able to answer some of your questions. So thanks everybody for such lovely slides.
So I'll stop sharing.
Thank you so much. So feel free to use the hand raise function if you have a question, or you can type it in the chat. I'm happy to start off with a few that came to mind. The biggest one sort of that we hear a lot is around timeline. So from this point where we're working in models to translation into individuals, what does that look like? What is the timeline there?
Liz,
looks like Liz has thoughts.
I was just going to say I could answer that, try to answer that, and I think the reason why we all paused is because we all care a lot about the answer to this, but it's a very it's a very wobbly answer. So this idea of drug repurposing, where if a drug is already used in people and can now be used differently in the context of cloves or prose, is really a much more rapid way to get something into clinical trial or use as compassionate use. And actually, I've had some experience of this making zebrafish models, again with Rob and a colleague, Veronica Kinsler, looking with BRAF mutations, where there are BRAF inhibitors currently available for cancer that could then be used in patients. So as Rob was saying at the beginning, there is sort of this ideal situation is that we could rapidly take a currently known drug and try it in a new context. As soon as you're making new drugs, and this is also something our lab does, again, in the context of cancer, it does take a lot longer. And that's because you have to, not only do you have to do all the science behind it, but you also have to ensure the safety of it. So that one can take much longer and is much more prone to it can be 5 years, it can be 10 years. That one is a very much harder answer.
Liz, we had
Rob, did you want to weigh in on that? You look like you were unmuting.
Oh, I just sort of have that look about me. No, just I completely agree with Liz. So, and the strategy has to be to lay your bets so you have the possibility of doing all those things. So in our work, we will be looking to see if we can find a smarter and better way or time or dose to use the medicines we have already. But at the same time, we'll be looking very hard for completely new things. So we don't have to choose between these options. We're going to try to do all of them to find the ones which work best.
Thank you.
Can I just add one thing? Because I'm sort of the other basic scientists, that's what we call them, basic, although I always find it a bit offensive when I say basic scientists, so fundamental scientists. I think something that's really important to emphasize as well is that before we can, it's tempting to just go and test a new drug or do a big drug screen, but really what's important is to understand the complexity of what we're dealing with. And I think for a long time, we haven't actually been looking into that complexity properly. And so that's something that we're trying to address as a team, taking advantage of the zebrafish model, but also the pluripotent stem cells and generating all these different cell types to try and understand what does PIK3CA actually do in each one of these cell types, as opposed to just assuming that it does the same thing in every single one of these cells. And I think that's really important because I'm sure many of you on the other side of the call will agree that whether if you have a muscle that's affected versus a blood vessel, you're experiencing very different things. And so I think capturing that complexity is really important in our research and then moving on forward from there. So taking the time to do this properly, I think it's really important, although it is tempting to obviously rush things. I think we all agree that at the end of the day, that's actually more a waste of time and money and false hope potentially. So yeah, it takes time, but we'll get there eventually.
Right. I do think that we answered Magdalena's question around, are we currently testing for new treatments or repurposing? Both, which is great.
I could just explain a little bit about how we will do that. So I completely agree with everything Ralitsa is saying, and we need to be really smart and precise in what we're doing. However, one of the advantages of zebrafish is they are, as we call them, cheap and cheerful. So they're actually quite easy to use, and we can test quite a few drugs on them. So now that we have this really great model, we will use the currently available drugs in our fish model to see if they're effective. And I guess one of the things we really want to understand is when to use them during normal developmental timing. Do you have to add it before the growth happens? Can you add it once the growth has happened? So there's sort of that element. And then the other thing about the repurposing is there are what we call drug libraries available, and we have those at our university, which are all the drugs that are currently available. And we can then start to put those on the zebrafish. And so it's called a screen, because we're sort of looking through things. But it has that potential to give us something new, but at least our starting point is something a drug that's currently available. So that's something that Hannah and I would love to be able to do, and we have the ability to do that in Edinburgh.
Yeah, so just to remind folks for some history, like the first goal of this zebrafish project was making sure we could create the model. So now the model is created, and so now the question is, what's next? And is our organization going to help to fund what is next? So we've checked the box for the first goal, and now there's a whole horizon of opportunities for us to consider.
Right. Moving on to the next question. So in the chat, we have one from Robert. And although we cannot give any specific medical advice here, the question is around which drugs are most effective and what are the side effects. I'm just going to add to that. How do you test for those things, right? So we're screening the drugs to see if they're effective in zebrafish. How do you decide on which ones to do and noting what happens after?
Okay, so the question sounds more clinical. So I'll start off with that. I mean, and my answer would be partly that it's many of the people on this call who can tell you about the medicines in cloves, because at the moment we're waiting for the first big trials that compare medicines against placebo, because you may well have heard of medicines such as alpelisib, which is the technical name for Vijoice. So one of the medicines which is now licensed in America, but not yet in Europe. And early on, it became clear that was an interesting thing to try because it targets the problem in cloves. And what we know so far after last 5, 6, 7 years is in some people that seems to do a lot of good. In some people, it doesn't do much good. But we can only know for sure how much good that is and exactly what the side effects are when that's been compared in a trial. And the best trials are the ones where we as doctors don't know what the medicine is, and people who are receiving them don't know what the medicine is, because we all want them to work. And if it's not double blinded like that, you can the results can be twisted by hope because you all want it to work. So we've seen enough to know that there are some medicines that certainly do some good. We're waiting to hear exactly how much good they do. But that also leads on to why the fish are so valuable, because to find out that answer is taking a lot of effort and a lot of money and a lot of years of people going along to hospitals and having measurements and having these drugs. So to really prove what is best takes a long time. And that's why it's so important to choose the best options right at the start. And the fact that we can take the fish, as Liz said, and the cells, and we can test lots and lots and lots of medicines, and we can take risks in those fish and those cells, means that we get to the most promising starting points faster. So as to what the best treatment is, that's very, very personal. It depends exactly what the problems any particular person with cloves has, and that's something they should be discussing with their own doctors. We know there's some medicines that do some good, and we're still waiting to hear the full story.
Thank
But
you.
we're impatient, and I'm sure you're impatient, and that's where the models come in.
So I have another question, and it's around what can patients and families do? We talked a little bit about providing funding, but what else how else can they contribute to the research? Ralitsa, you look like you had a thought.
I was actually going to pass it on to Rob, but because I was thinking about the registry, but Rob is probably the person to be talking about that, not me, as I'm not clinically qualified.
So I'm worried about taking all the airtime here. So there's lots and lots of things. So first of all, the models are really good. They're ways to accelerate progress, but in the end, they always have to be carefully checked against samples from people with cloves, and they have to be what we find in our models. We only know if the models are good if we can compare them in detail to actual experience of people with cloves, and that's only possible if people are happy to share their information in a registry and sometimes their samples. So that's one thing, making sure that there is as much information about the thing that we're trying to treat available as possible. And then also as researchers, we desperately need to know what is most important to you all as well. So we want to be working on the things which are most important. So working together to tell us what the priorities are, which bits of the condition are most worrying or bothersome is very valuable. And then when, of course, money we've talked about already, helping us to get the initial studies which are needed to persuade funders to put more money into this, and also being prepared to write letters of support when we put in grants to explain to the people who give out the money that we are working in partnership with you and we're not just doing this in isolation. So there's lots and lots of things to do. It's a partnership, it really is. And sometimes there are types of funding which you apply for as a patient group and we give support for. So it happens both ways round.
I just wanted to, oh, Ralitsa raised her hand. Okay, well, hold on. So I just wanted to, we all have like this view of what researchers do. And a lot of times it is like in the lab with some Petri dishes and, right, Ralitsa's like nodding because I just saw a video about this. But I would love to hear like from, I know Ralitsa probably has a story to tell, like, of like why interacting with families and people who are impacted is important, or do you have a story to share about why people should be talking about this with researchers, right? So just like, why is it important for our community to continue talking about this, talking about how hard it is to live with this condition and trying to like bring more people in? So does anybody want to riff on that?
Okay, you're nodding. Good. Well, one good example was actually last week. I was approached by a very senior researcher telling me I was at a meeting in Europe listening to some talk about treatments of PROS, and I heard that it is cured. So that's what I was told. And I said, is it cured? Well, then my research is obviously, my job is done, right? So I think it's really, really important to continue to raise awareness that this is not cured and there's still lots of questions to be answered. And it's really important to address those questions and to also be very open about the limitations of current treatment, current research as well. So that was actually going to be related to the other point that I wanted to make. I think you as a community absolutely are entitled to hold us accountable. So you are giving us money, but you also have certain expectations in return. And I think some of those expectations are for us as researchers to be sharing our results openly, to be collaborating. And obviously, when you're doing research as your job, there are certain expectations about formal papers, etc. That stuff can take a lot of time, and that's not necessarily what's going to help you here and now. And you really should be pushing us to be much more collaborative and much more open about these things, and not having to wait to share results through some of the conventional routes. And Kristen and the close community have been organizing this fantastic roundtable that we've been running for the last 2 years now, with bringing together researchers from across the world working on PIK3CA in various ways, and sharing results with one another before any of those, before they're released through those formal communication routes. So I think that's really important, and we've had several members of this community actually attend those talks. They're very jargon heavy, they're meant for researchers to share results, but it's really nice to see you attend once in a while and also hear your stories. And it kind of keeps us grounded in terms of remembering that at the end of the day, this is money that's been given to us to make progress that is benefiting you. And so that's really important, that engagement with the patients. And towards the end, hopefully there'll be some time to introduce another more junior addition to our research team, and Olivia, who hasn't been introduced yet or hasn't introduced herself yet, will perhaps give you another insight into why it's so important for us to be interacting with you and how much that's motivating us on a daily basis. That was my answer mostly. But yeah, keep raising awareness. That's really important, because this has not been cured, and I think we can all agree on that.
And of course, I'm going to plug our registry, which you can find out more about on the closed website, where you can provide details on your disease and disease progression that can be accessed by researchers to move things forward. Okay, I have a zebrafish question. I'm just going to read it out. There are a number of point mutations of PIK3CA that have been implicated in pros and cloves. How does the zebrafish and cell model account for this? Have you selected one mutant to use as a model to represent all the various mutations of PIK3CA?
So I think I can jump in with this one. So I gave you a summary of what I'd done, and I intentionally kept, the DNA that I injected in vague for simplicity. But one of the advantages of the system I'm using is that it's sort of like 3 jigsaw pieces stuck together, and I can swap those out. So what I've been able to do is to test 4 different variants. So these are the hotspot mutations. So I've tested 1047R, 1047L, and the 2 helical domains. And so I've injected all of those into the fish, and I was wondering whether they would have the same sort of effects, and actually they do. So I've been trying to score the different phenotypes I see. Yeah, so I'm looking at more than one of them for sure, and it is one of the advantages of zebrafish that we can do that relatively easy. I know it's difficult, but more difficult perhaps when you're breeding, mouse lines, to be able to have that many going on at once. So I'm currently using all of them for the sake of publishing a paper. I'm focusing on one, but that's because I've been able to test that the others are behaving in the same way.
Hannah, are the helical domain, is that 545?
Yeah.
Okay. Is it 2 545s or 542?
So there's an E542K and an E545K.
Thank you.
And also, I should say, because my system relies on just activating these mutations everywhere, what I've also done is to put an ordinary copy of PIK3CA in, just to see if it's just the problem with my injection, and that doesn't produce the closed phenotype, so I know it's specific to the mutation. I think Liz has raised her hand.
About Scott's question about Vajoyce, and I'm sure that Rob may be able to answer that in more detail about does it cause other effects on growth. one of the things we do look for, one of the advantages of using a zebrafish or a mouse or a whole animal system is that we can look both at the effect on the diseased tissue or the overgrowth tissue, as well as on the healthy tissue at the same time. And so that's something, I think that's a really good question about, you know, how do you develop drugs that target exactly what you want without causing harm on the healthy tissue? And that's something we'll also be looking at in our models.
Rob, I see that you dropped some things in the chat. Did you have anything you wanted to add?
Yeah, so it was just a clinical comment that really, theoretically, more than anything, there could be an effect of VJOICE on growth and puberty timing. I haven't seen any reports of that. I don't personally know of any evidence for that. So if there is any effect, and I don't know if there is, maybe it'll be very small, and it'll only really be from the big trial that's happening at the moment that we'll know for sure. So they're measuring that very carefully indeed. But just exactly as Liz said, rather than waiting years to know the answer, we can get a sort of preview of that by looking carefully at models such as the fish, and we can try things like high doses for short periods of time, low doses for long periods of time. We can go through lots of possibilities that would take years and years and years to test one by one in people. So
thank you. We have another question in the chat, and it's sort of to all of you. So how did you get involved in this research? To begin with, it's, you know, a small community. And then sort of, are you in contact with other researchers? What does this sort of community look like?
This might be a good opportunity actually to hand over to Olivia in a second, but first and foremost, I think a lot of us have to thank Rob for being involved in this research. As he mentioned already, his group was one of the first to discover the PIK3CA mutations can cause these types of overgrowth disorders. So certainly by serendipity, I ended up in his lab, and so that's how I ended up working on this. And I continue, and I'm particularly interested more generally in everything that PIK3CA does, beyond also pros and cons. And so we're part of a larger community of researchers with whom we share our research. But I think it will be much more interesting to actually hear from somebody like Olivia, who is going to be starting her PhD in my lab September onwards this year, because she's probably very much in the positions that I was in 10 years ago when I first started. And it's easy to forget that, but yeah, the excitement that comes from being involved in this type of research and being able to make a difference is probably best conveyed in her own words. So Olivia, I'll hand over to you to say a few words about why you're involved in this research and what it has meant for you in the last 6 months.
Okay. Hi, everyone. Maybe I'm gonna actually share my screen because I prepared like sort of a summary slide that maybe is gonna make it a bit easier for you to visualize. Let's see. Why? Okay, there we go. I hope everyone can see that. So again, hi everyone. My name is Olivia, and you can see me right here. And I'm a student and technician at the Madsen Lab. I'm very happy to be here with you today, and I just wanted to quickly give you an overview why I'm involved and what it means really for me to be in this space. So just to start from the beginning, I started in the lab around maybe 10-ish months ago now, I think. And I very quickly realized that there's a ton of work to do, but I actually really enjoy it because every time I'm in the lab, or even when I'm not in the lab, actually, I'm learning something new every single day. And so since I'm a fairly new addition to the PI3K signaling field, thinking about what I'm going to say here today, I started thinking about when I first started and how scared I was actually to first join. I was actually very afraid to touch anything in the lab because I kept thinking, you know, after all, I was very conscious about the fact that, you know, we're very privileged to do this research, and I really didn't want to break anything or or waste any funds. So yeah, I was very scared at first, but I think the support that I've gotten from Ralitsa and the team and the community as a whole, I think it really has helped me out. And ultimately, joining this lab, I think made me realize how important this is. And because also of this very supportive community, I decided to stay and follow further with my PhD. So I just gave you a very quick sort of slideshow here, just so you can see what we see on a daily basis. A lot of what you can see here is looking at cells, taking care of cells, again, looking at cells. And that's because, as Ralitsa mentioned earlier, we generate these models with the mutations to understand really how the cells look like and how they differ from the normal cells. But yeah, in the end, as I mentioned, I think what has been really, really important for me, probably the most important part, is the support from Ralitsa for welcoming me into her lab and mentoring me. Then there was Gray, who was a previous student of Ralitsa's, and she was there just as I was starting out. So she really helped me with the confidence and sort of following through and learning all the techniques that I use every single day now. Also, the MRC-PPU, which is the research center that we work at, which has been very supportive with our research. And I think probably the most important would be the close and the pros communities. And that's just because I feel like I have gotten to only very briefly meet some of the people, but the encouragement and the support, it has really been overwhelming. And I really feel like it's such a welcoming and fantastic community, and I love how much collaboration there is. So yeah, I think just to wrap this up, really, I just wanted to say that it really means the world to me to do what I love and contribute to the new knowledge while being in such an environment. And with that, yeah, thanks for listening.
Thanks, Olivia. I should also say that Olivia was actually supported partly by funding from Caravan, which is another organization, a patient, or sort of a rare disease supporting charity that actually helped with some of the funding, and that was in part thanks to Kristen and Noreen for putting me in touch with Caravan to make sure that Olivia could actually work in the lab and stay on. It wouldn't have been possible actually without that support. So again, it means a lot to us that we have all of this support from you guys.
Thank you, Olivia. Thank you, Ralitsa. So we have 2 minutes left, and I love Adam's question. So I think let's end on Adam's question, knowing that this is just the beginning of a conversation that will continue on and on. Noreen, do you want to read Adam's question? And we might not be able to answer this question in the moment, Adam, but it's something that I know the board of CSC is really interested in, and knowing kind of how to keep the momentum going. So I'm going to pass it to Noreen to read, and then we can see if anybody wants to answer.
Sure. So starts off, I'm interested in the longevity of this research. The success in a year or so of initial building the model to where we are today is amazing, which I think we can all agree with. Is there a known monthly sort of burn rate, spend rate of the dollars, pounds, euros, and how much financial support would be needed to move the research forward?
Depends really what you're doing. Yeah, well, I'll hand over to Liz for the zebrafish in a second. But for example, the pluripotent stem cell work, in fact, Olivia may know the numbers better than me because these days she's doing the ordering. But we're talking about 3,000 to 3,000 pounds just a month, really, when it comes to taking into account all that's required for taking care of the cells, making images, analyzing, and that's just mostly consumables. On top of that, you obviously have people's salaries as well. So it can quickly get expensive, and it's not something that a charity like CSC can fund in its entirety. But where the support is really helpful is pump priming, really, which is how you guys have been helping us so far. I'll hand over to Liz, but the zebrafish, luckily, are more cheap and cheery.
So Ralitsa is correct that it's really the strategic funding that's so important for us. And so with the funding that we've had from cloves, we've been able to keep Hannah in the lab and hire Hannah to do this project. And that's really where the skill and expertise comes. And so if we want real numbers, about 120,000 would keep us going for another 18 months. And that would allow us to do something very strategic that we would develop in consultation with you. For example, do we want to do a drug screen? Do we want to understand more about those pink cells? You know, what are the priorities? We can always work with less, but it is difficult. Rob mentioned about the challenges of having a scientific career, and where we really get momentum is when we can say to somebody, you know, we can commit to you for 3 years, or that kind of time frame, and then people can really go deep into the work and get it done. So I will say that that would be a wonderful number for us. Our job is to go get more funding. And so exactly what Ralitsa said about the pump priming, with the funding you've already given us, we've been able to make this model, and with Rob and Ralitsa and Hannah, we've put in a grant application for £650,000. So really, we see your money as absolutely critical to helping us, empowering us to get to the next stage so that we can go to some of the big funding agencies and get big investment that way.
You might not realize this, but one of the biggest risks to any research in any disease is that most scientists don't sit in secure jobs for the rest of their career. Eventually, universities do say, look, we'll stick with you for the rest of your career by the time you're old like me. But for many, many years up until then, people are only employed when they're able to attract in funding from outside. So that's often for 2 or 3 years at a time. So one of the biggest risks is that there are gaps in that time, and people who've invested time and effort to do really good work are then lost because that there isn't extra money to either get up to steam in the first place, which we've done beautifully, or sometimes to cross small gaps which occur in funding as well. So it's a very insecure career path, at least for the first 20 years, and it's the people who are most important to keep in there. So.
Exactly, and we also call that funding bridge funding, and it's really to help bridge us until we can get those larger funds to give security for the scientists like Hannah.
Thank you all. Thanks, Adam, for asking that question. I'm just going to say thanks to everyone, and I'm going to pass it over to Lauren to see. So anyone here who's passionate about funding research, we have an initiative that's happening right now where you can take an active role and do that. And now I'm going to be quiet and get back in the back seat, and Lauren is going to explain what that opportunity is. And thank you to all of our presenters and for being here today.
Yeah, I want to start off by saying, I think just listening to all the scientists talk about the greater understanding we have of what the PIK3CA gene does and what it does when it is not doing like the average thing, the steps we've taken to understanding that and having that model, just it gives me so much hope. And so thank you guys for that and for sharing that with us. And I can't wait to hear what comes next. What comes next for our community, and what I'm hearing from the scientists is being able to help with bridge funding, right? Getting everybody going, and then being able to bridge the gap between what you've done and being able to get these huge amounts of funds to let you keep going. What we are doing right now is our $1 million bike ride campaign. All of the money we're raising through this campaign will be earmarking. I keep saying that word wrong, so I'm like making sure it's not dog earing like what you do in a book. Sorry, earmarking for research. And so we are trying. Our goal this year is to raise $25,000 through this campaign. You don't have to ride a bike. You can walk, you can wheel, you can run, get your friends and do a thing. I run with a group of women, and I'm trying to get us all to let's all do a run together. and raise money for Clove syndrome with that. So it can be casual and what have you. But check out our website. We're posting on social media about it. We have a cute little set of reels coming on Instagram about our zebrafish mascot that we named Zeke. So yes, please help us out, spread the word. I know our community is small, and I'm really cognizant of the fact that we are asking for help from a very small pool a lot of the time. And I don't want to make everyone feel like what the ask is to you individually, right? Spreading the word and finding creative ways to help us fundraise, and even just helping us get the word out is just as valuable. So that. Is there anything I missed, Kristen? I'm sure there is. I mean, it's not really early where I am anymore, but.
No, just, if you have questions, reach out to us and
Please
ask
do.
questions. And we're here to help you set up a fundraising page, create a story, talk about why research is important, all of that stuff like that. If you've done that a few times, you know how to do it. If you're new to it, it can be a little overwhelming, but we're here to help. And we are all really motivated to find options for our loved ones, especially those of our loved ones who do not have options. So hi, Franco family. It's nice to see you. So let's do this together. Let's join forces and make this happen. So
yes. Yeah. And if you ever want to brainstorm ideas, hit me up. I'm happy to help with that.
And we will put this on our YouTube page, and that will probably happen in a week or two, and we'll make sure that it'll go out in a newsletter post, just that it is live for folks who want to watch it. So thank you all very much. I think we're complete.
Yeah.
Thanks to the Research Dream team.
Thank you, everyone.
Your support.
We really appreciate being able to speak with you today.
You're all welcome to unmute and say goodbye if you'd like. I started attending meetings where people do that, and it's like one of my most favorite things. So if you want to unmute and say goodbye, here, it's time now.
Goodbye.
Thank you very much.
Thank
Bye-bye.
Thank you.
Bye,
Thank
everyone.
you. Bye.
Thank you.
Bye-bye.
Have a good one.
All your hard work.
Yes. Thank
Thank
you.
you.
Bye, guys.
Bye.
Great. Okay. Good night, everyone. Thank you so
Love it
much.
to you all.
Bye. Bye.